All right. Well, thank you very much for this opportunity and for the prestigious award. Well, my lab is using single molecule biophysical tools to study nanomachines. So we basically heard a lot about nanotechnology. Well, we're just trying to make devices smaller and smaller, try to do robotics, make small machines, maybe like to try to fit a lot of devices into the tip of a needle. Well, these are great efforts, but what's really impressive is that the nature already has nanomachines that are functioning in our bodies and that are far more sophisticated than what we can produce. So my lab is trying to use methods that we can actually manipulate, like this mafia thing, manipulate nanomotors and try to understand their mechanism. So what type of motors I'm talking about? Well, for example, this example is ATP production machinery called ATP synthase, which we basically eat food, and then we basically rip off the hydrogen ions from the food, and then that hydrogen ions pass through this channel. As these ions pass through the channel, they actually rotate this basically turbine. That turbine is actually then basically makes ATP for us. It's cellular fuel. Well, basically in this process, pretty much like basically a hydroelectric power plant, that we basically take a chemical material like food, convert it to electrostatic energy, then to mechanical energy, and then to the useful energy. So this is basically occurring in our cell. This is one of actually a very impressive machine that actually can rotate around 90,000 RPM, which is roughly about 10 times faster than a Ferrari engine. Pretty good. Yeah. So another type of motors are linear motors. These guys actually walk. And they actually transport materials that are basically 10,000 times larger than their volume. And this is actually very important because our cells are very big. We need to distribute material around, and we cannot just leave it to diffusion. And they basically they produce force, transport material, and also produce force for other processes such as cell division and cell motility. Well, I'll talk about these motors actually in my talk. So there are actually 3 types of motors that function along what we call cytoskeleton, which are basically linear tracks that function as highways and railroads inside our body, inside our cells. There are 3 types of these motors. Basically, I'm not going to go through their structural detail, but what they are is nature's smallest bipedal walkers. Their job is to walk and carry stuff. So we were interested in how they walk. Well, there are multiple models of walking, but I will only highlight 2. One is basically what's called hand over hand, which is pretty much human walking, that basically motor walks by taking left and right alternating steps. Okay, this is a walking model. The other way to move is inchworming, which is basically a baby crawling model, where one of the feet is leading and other one is basically just dragging along with it. Well, how can we actually, this is now an example of basically a scientific discovery, how can we actually distinguish between these 2 models? So if I am walking at the dark, and you cannot really make out how I'm basically walking because you need to see my feet, but we cannot see the foot of the proteins because they're too small. We have to label them with a sensor, fluorescence sensor. Pretty much in the stark darkness, I basically label one foot with red bulb and other foot with a green bulb. If I can see the pattern of translocation, movement of those bulbs, then I can actually make out how they walk. Well, you can do this to humans. Doing that to proteins is far more difficult. Because small molecules cannot be observed at their sizes. If the size of the molecule is smaller than the wavelength of light, which we just heard about, then they are limited by the size of their wavelength. They look much bigger. Basically, the size of the protein, if this is a typical image of a fluorescent protein, let's say, the size of the protein is this laser pointer, this guy. Okay, very small, but this is how big you see it, right? To be able to see it really, to be able to see it with very small, with a very good resolution, what we have to do is that we have to basically collect a lot of signal and then determine the center, which represents the position of the molecule. Then we can actually achieve required resolution to be able to see how they walk. So this is actually what we have done. It was basically a very simple idea, but the challenge was that basically we are labeling a single protein with a single fluorescent dye molecule, which is very, very dim. So you don't get enough signal, it just bleaches right away. So we have found ways to improve this by 100-fold. Now we can basically these are the individual dye molecules. We can take their image, feed it to 2-dimensional Gaussian, and get 1 nanometer localization. nanometer, which is 1/billionth of a meter. So if you make the world billion times smaller, it is at the size of a soccer ball. So billion is a big number. All right, so if it's 1 nanometer localization ability, now we can basically, now I can see how proteins walk. So we labeled a protein called myosin 5. This is a single protein that's walking along a linear track on the surface, which is not labeled. You only see the protein. And we can actually see the stepping size of the protein. Myosin 5 protein, its center of mass takes 37 nanometer steps, but its feet are taking twice the center of mass step size. So basically, to an analogy to human walking, if my body moves 30 centimeters at every step, my right foot will take 2 times 30 centimeters. It will be 60, and the next step it's going to stop to let the other foot to take a step. So if my body moves 30 centimeters, my feet will take 60 alternating steps. That's exactly what the protein is doing. Its body is taking 37 nanometer steps. Its feet are taking 74 nanometer steps. So it works like humans. Well, I mean, we can actually do it with better now, but we can actually label another protein with 2 different colors. Now, this is my real example where the person walks in the stark darkness with red and green lights. And then we can actually see that the 2 feet swap paths. This is position versus time. So first the blue foot takes a step, then the red, blue, and red, and so on. We can actually see that it walks by taking left and right alternating steps. So getting back to this movie, which was made after this work, it basically walks like humans. And this mechanism actually tells quite a lot of things about the mechanism of this movement, which I don't have time to explain. Let me actually turn our attention to another protein called cytoplasmic dynein. Well, dynein is actually a very big protein, nature's biggest peptide known. And then this protein has a very different mechanism of motility. So we basically were thinking that maybe all the proteins walk like humans. Then we actually did this experiment. We labeled them with 2 different colors. And you can actually see that they move on the surfaces. Then we actually looked at their motility. Well, they don't walk like humans. Their 2 feet are independent of each other, but they can still walk. Well, I don't have a really nice animation for that, but I have a movie for that. So Monty Python comes to my help. Misregistered steps, sideways steps, and so on. So it's walking either of these guys, basically. Then we were interested in how do these proteins know to go in one direction? Because basically, these tracks are like highways. So there's basically 405 south and 405 north, right? So the cargoes need to go in one way, and they need to be able to get back. The way things work in cell, there's one track, not two different, basically, south and north. There's one track, but some proteins walk forward, some proteins walk backwards. In the study of other motors besides dynein, the mechanism was that there is a first-generation mechanism that these proteins chew up ATP, cellular energy, and then they produce force in one direction. If you reverse the direction of this force production by engineering and new constructs, then they start walking backwards. Okay, we did the same experiment to dynein, expecting that it would do the same. If I can reverse the force production, it will start walking backwards. Well, dynein force production machinery is this. Well, let's just wait for a second. I'm just misregistered with my movie. All right, so the idea is that ATP comes in, binds to this head, and it creates a conformational change which moves it forward to this direction. The left is forward in this case. Okay, well, this is actually very easy to change from our perspective because we can actually, this foot that actually lands on microtubule filament is actually a helix that it actually rotates around its own axis. If I add half a turn, then protein just basically reverse 180 degrees and should really walk backwards this time. Well, it doesn't. This protein produces force backwards, but still walk forward. That was a really confusing moment for us. So we tried to understand how this can happen. So we then checked, what is the thing that we didn't change? The only thing that we didn't change is this small domain that binds to the track. So that must basically, that's creating asymmetry to break the symmetry of diffusion. How many minutes? Half a minute. Whoa. All right. Well, then I can only show my movie. All right, so we discovered that these foot are asymmetric. They're not made to walk backwards. They can only release forward. So it's like a ratchet. So basically, the way it works is that when the protein binds ATP and diffuses along its track, it can only bind in this basically forward-depending angle. and when the force is being produced between two ends, it only releases forward. So it releases forward and binds in the forward direction. So it doesn't really matter which direction you produce force. You basically, ATP is only used to bind and release, and then basically the protein is made to bind Walker by its foot. So it's basically a very different design for this protein. Well, I think I'll stop right here, and thank you for listening.